Hello, everyone. My name is Hao Wang, and today is December the 16th, 2025. In this presentation, I'm going to explain how the gastric phase of digestion is regulated. And I'm going to keep the focus on how the digestive system communicates and coordinate its actions. So, what is the gastric phase? So the gastric phase begins with the food enter the stomach first, and the stomach is not just a bag that stores food; it's also a control center that adjusts secretion and movement based on what is inside. The big goal are just simple. The stomach needs to increase secretion, especially acid and amines, so digestion can stuff efficiently. And needs to increase mixing, so the meal turns into chim. And it needs to control emptying, so chim enters the small intestine at a safe pace. Over the next slide, I will walk through what triggers the gastric phase, how neutral reflex coordinate responses, how hormones reinforce the message, and how feedback signals prevent the system from just going too far. So, what triggers the gas phase? To understand regulation, we first need to ask what starts the gastric phase. So the stomach responds mainly to two kinds of information, mechanical information and also the chemical information. The first is the mechanical stimulation. When you eat, the stomach will stretch. That stretch is detected by receptors in the stomach wall. This is like the stomach saying, oh, there's volume here. I need to turn on mixing and secretion. Second, it's chemical simulation. So as the food begins to break down, the stomach sends peptides and amino acids in the lumen. These are strong signals because the stomach is designed to begin protein digestion early. Also, right after a meal, stomach pH can rise briefly because food buffers acid. That change helps actually support activation of gastric activity. So the key idea here is communication from sensors. Strength receptors and chemo receptors collect information about what is inside the stomach, and that information becomes the trigger for coordinated responses. Now let us talk about neural control. This is where coordination becomes very obvious because the stomach uses reflex pathways to respond quickly and in an organized way. First are the short reflex. These are local reflex controlled by the enteric nervous system, or we just call it ENS. So the ENS can detect changes in the stomach wall and generate responses without needing the brain. This allows actually fast local control of secretion and motility. The second thing is lung relaxes. So in the stomach, the most important lung relax is vagal reflex. Sorry for my pronunciation, which involves vagus nerve and brainstem. So signals travel from the stomach to the brainstem, then back to the stomach. This helps coordinate stronger and more widespread responses when a meal is present. And the FM match what we want during the gastric phase. So the neural signals increase acid secretion from peripheral cells, increase also the pepsinogen release from chief cells, and increase gastric mixing and motility. So the message of this slide is that the nerve system is acting like a coordination network. It takes sensory input from the stomach and turns it into organized output across glands and muscles. After neural control, the next layer of communication is chemical signaling. So hormones and paracrine messengers help amplify the response and just fine tune it. So a key hormone here is gastrin. So gastrin is released by the G cells. It is stimulated by peptides and amino acids in the stomach. It is also supported by vagal imports, which shows how neural and hormone signals work together. Another important message here is histamine. So histamine is released by ECL cells and acts locally. So it strongly boosts the acid secretion. So here's the main coordination points. So parietal cells respond best when three signals are present together: the acetylcholine from nerves, the gastrin from G cells, and the histamine from ECL cells. When these messages arrive together, acid secretion increases much more than with any single just signal alone. But the stomach also needs breaks. So these cells release somatostain. So somatostatin reduces gastrin release and reduces acid secretion. This helps prevent excessive acidity and keeps the system balanced. So the key idea here is that the regulation is not just turning things on, it's turning things on in a more controlled way with built-in inhibition. Now that we have the control signals, what do they actually produce? This slide focuses on the outcomes that are coordinated during the gastric phase. So the first is the secretion. The parietal cells increase hydrochloric acid. Acid actually helps denature the proteins and creates a right environment for protein digestion. So the chief cells then release pepsinogen, and in the acidic environment, pepsinogen is converted into the pepsin. Then the pepsin begins breaking protein into the smaller pieces. The second thing is motility. So the stomach produces mixing waves that turn the meal and blend it with the secretions. This turns the meal into chunks. This mechanical mixing is essential because enzymes works better when food is evenly mixed. The third thing is controlled emptying. So the pyloric sphere actor regulates how fast chimps enter the duodenum. The stomach does not dump everything at once. It actually coordinates mixing and secretion while delivering chimps in small portions. So overall, the stomach is coordinating secretion and movements as one integrated process, not as separate actions. So this slide explains how the system knows when to slow down and how the stomach coordinates with the small intestine. So the first is feedback inside the stomach. As acid secretion increases, stomach pH actually drops. So low pH activates these cells, which release somatostatin. Somatostatin reduce gastrin and reduce acid secretion. This is a classic negative feedback loop, and it protects the stomach from become overly acidic. The second is feedback from the duodenum, which is a part of the small intestine. When chyme enters the small intestine, the duodenum monitors it. If chyme is too acidic or contains significant fat, inhibitory signals are sent back to the stomach. The purpose here is protection and pacing. So the small intestine needs time to neutralize acid and process nutrients safely. So the net effect is that gastric emptying slowly when the intestine is not ready. This is a great example of organ-to-organ communication, where the intestine can choose the pace and the stomach adjusts accordingly. Finally, I want to connect this regulation to a quick clinical example, then summarize the main points. So a common clinical issue involves excess acid and mucosal injury like peptic ulcers. When the balance between acid production and protective effects is disrupted, the lining can be damaged and symptoms can appear. So treatments also often just focus on reducing acid outputs, which helps restore a safer environment for healing. Now let's summary. So the gastric phase begins when the food enters the stomach. The distension and chemical signals like peptides and amino acids activate the sensory receptors. The neural pathway, including ENS short reflex and vagal long reflex, increase the secretion and motility. Then the hormone and paracrine signals, especially the gastrin and histamine, amplify the acid secretion, while the somatostem provides inhibition. Together, these signals coordinate acid production, enzyme activation, mixing waves, and controlled emptying through the pyloric sphincter. Finally, negative feedback in the stomach and the inhibitory feedback from the duodenum prevent excessive acidity and slow emptying to support the transition into the intestinal phase. Okay, that's all. Thank you for watching.